Hello everyone, welcome back once again to my educational channel on biology. Teacher Janet here. In this video, we're going to discuss subtopic 4.4 in the SPM biology from 4 syllabus. The title is Types of Lipids. So this is the last video, the part 3, third video and final video on lipids. In the second video, we've already discussed fats, alright? So we're going to talk about the functions of other types of lipids, which are the phospholipids, waxes, and steroids, which are all found in the human body. Alright, and they are very important. They have different roles and functions and are essential for the health of the person, okay? Health of the human body. So for example, fats. Do you know that 65% of our brain is made of fat? So fat is very important for brain functioning, for proper brain functioning. Also, the type of fat that we eat must contain the omega-3 fatty acids that are used to build the brain tissues. So these types of fats are important for learning and memory. Let's find out more about the other different types of fats like phospholipids, waxes, and steroids. The learning standards for this lesson are as follows. 4.4 Lipids. First, we should be able to explain the different types of lipids. Secondly, justify the importance of functions of lipids in the cells and multicellular organisms. And thirdly, describe a food test to test for the presence of lipids. So let's recap what we have studied in the previous video. All right, and we're going to also explain the 4 different types of lipids that are in our syllabus. Now, we have already studied about fats and oils. They are called triglycerides, okay? So they are formed from one glycerol molecule combined with 3 fatty acid molecules. This is for one molecule of fat, okay? One molecule of fat contains one glycerol molecule and 3 fatty acid molecules. And then fats are divided into 2 types, saturated fats and unsaturated fats. In saturated fats, the fatty acid molecule is saturated, meaning that the fatty acids have only single bonds between the carbon atoms. C, single bond C, right? So they cannot react and form any more bonds with other hydrogen atoms because they are already saturated. The bonds are already fully used to bond with other atoms, all right? One carbon atom can bond with 4 other atoms. Now, for unsaturated fats, they contain fatty acids which are unsaturated. This means that the fatty acids have at least one double bond between the carbon atoms. And when this double bond breaks and becomes a single bond, then the carbon atom can form new bonds with other hydrogen atoms, right? So that's been discussed in the previous video. Now for phospholipids, they are different because each phospholipid molecule consists of one glycerol molecule, 2 fatty acid molecules, and one phosphate group. That's why it's called phospholipid, right? So basically, phospholipids are used to build the plasma membrane and to allow the movement of certain substances across the plasma membrane. For waxes, a molecule of wax consists of one alcohol molecule combined with one fatty acid molecule. So wax is a waterproof substance that's found on the leaf, okay, in the waxy cuticle, and it helps to reduce water loss by transpiration. Now, steroids. Steroids do not contain fatty acids. Examples are cholesterol, progesterone, estrogen, and testosterone. Take cholesterol, for example. It makes the phospholipid bilayer stronger and more flexible. Okay, so the cholesterol is embedded in the phospholipid bilayer, and it helps to make the phospholipid bilayer stronger and more flexible. Here is a useful overview to show you the structure of the different types of lipids. So for fats and oils, which are triglycerides, each molecule of the fat is formed from one glycerol molecule and 3 fatty acid molecules as shown here. This we have discussed in the previous videos, all right? So the glycerol here is the alcohol, and then it binds with the fatty acids, one glycerol molecule to 3 fatty acid molecules. And when they bind together, 3 molecules of water will be eliminated in a process called condensation. All right, when the glycerol molecule and the 3 fatty acid molecules combine together. Now for phospholipids, they are different. Each phospholipid molecule consists of 1 glycerol molecule and 2 fatty acid molecules and 1 phosphate group. Okay, as found in the plasma membrane. Okay, so here we see 1 phospholipid molecule, and you can see here 2 fatty acid molecules attached to 1 glycerol molecule. So the difference between phospholipids and fats is that the phospholipids have only 2 fatty acid molecules, but the fats have 3 fatty acid molecules, okay, in each of the molecule. So for glycerol, for phospholipids, since it forms 2 bonds with 2 fatty acid molecules here, the one more bond is used to bind to the phosphate group, okay, at the other side here. Now for wax, a molecule of wax contains 1 alcohol molecule and 1 fatty acid molecule, okay. So this is not glycerol, it's just an alcohol in general, and alcohols, they have the OH group in them, okay, hydroxide group in them, or hydroxyl group. The fatty acid has the COOH group, which is the carboxyl group. So there's a reaction here, and a bond will form between the fatty acid and the alcohol, okay. So this is wax. For steroids, they do not contain any fatty acids at all. Examples are cholesterol, progesterone, estrogen, and testosterone. Steroids have a backbone which consists of 4 rings containing carbon atoms. And then there are other molecules at the side, which is not shown. Okay, but basically they have this steroid backbone that consists of 4 rings. And there are no fatty acids in the molecule. So here we see a pattern. Fats have 3 fatty acid molecules in each of its molecule, right? Each fat molecule has 3 fatty acid molecules. Then for phospholipids, each molecule has 2 fatty acid molecules. For wax, each molecule of wax has 1 fatty acid molecule. And for steroids, they do not have any fatty acid molecules. Okay, so remember the acronym FAT POWs. Fat, these are all the 4 types of lipids. Fat stands for the fats. POWs, P stands for the phospholipids. Okay, PO. And then W stands for wax and S for steroids. Okay, so as we go from one lipid to the other, the number of fatty acid molecules will become less and less, right? From 3 fatty acid molecules to 2 to 1 and then to 0. So remember fat POWs for the 4 types of lipids. Okay, just a quick recap of the formation of fats or triglycerides. So this was discussed in video number 2 on lipids. Now, a triglyceride is formed by condensation when 1 glycerol molecule and 3 fatty acid molecules combine with the removal of 3 water molecules. Okay, this is how a triglyceride molecule is formed. So what is a glycerol molecule? A glycerol molecule is a 3-carbon alcohol that has 3 hydroxyl groups as shown here. So 3 carbon atoms are found in the glycerol molecule, okay, here, and then there are 3 hydroxyl groups. All right, but for just alcohol in general, an alcohol is an organic compound that must have at least 1 hydroxyl group bound to the carbon atom. Okay, so alcohols have the hydroxyl group, OH group. As for fatty acid, what is in a fatty acid molecule? So a fatty acid molecule consists of a chain of carbon atoms, as shown here, with hydrogen atoms, all right, at the 2 sides here, along the chain, and a carboxyl group. Now this is the carboxyl group, COOH, at one end of the chain. So fatty acid molecules will have this carboxyl group at one end of the chain, COOH. So the first type of lipid that we'll discuss is the phospholipid molecule. Now structure. A phospholipid molecule is made up of one molecule of glycerol that combines with two molecules of fatty acid and one phosphate group, as seen here. Okay, so one molecule of glycerol, okay, and this corner here, and then you have two molecules of fatty acids below, and one molecule of containing the phosphate group here. Okay, functions. Phospholipids are a major component of the plasma membranes, as seen here. So we can see that the plasma membrane is made up of two layers of phospholipid with the tails facing each other, right? That's why it's called a phospholipid bilayer. There are two layers of phospholipid molecules. And here we see the cross section showing you the phospholipid molecule. So each phospholipid molecule has a hydrophilic head and two hydrophobic tails, as discussed in chapter 3. Okay, let's find out more. Okay, now we go on to the function first. So functions of phospholipids, they are a major component of the plasma membranes. They build up the plasma membranes. Okay, they form the phospholipid bilayer. And secondly, the phospholipid bilayer allows certain molecules to pass through it. For example, oxygen can diffuse through it into the cells for cellular respiration. Carbon dioxide is produced through cellular respiration can diffuse out from the phospholipid bilayer. Okay, and even water can diffuse through osmosis across the phospholipid bilayer into the cell. So looking at the structure of the triglyceride or fat molecule and compare it with the phospholipid molecule, they are quite alike, except that okay, a phospholipid has a structure similar to a triglyceride or fat, but with only two fatty acids instead of three, right? So for triglycerides, there are three fatty acids. For phospholipids, there are only two. In addition, the phospholipid has an extra group called the phosphate group. Okay, that's why it's called phospho, standing for phosphate, lipid. So phospho is this part, the phosphate group, and lipid is this part, the glycerol group or glycerol molecule combined with the fatty acid molecules. So looking at the phospholipid molecule here, we can rotate it about 90 degrees and then we will see the upright structure like this that resembles the phospholipid molecule that we studied in chapter 3, okay, on the structure of the plasma membrane. So the hydrophilic head is this part, the water-loving part, consists of the phosphate group attached to the glycerol part, okay, and it is negatively charged. It has one negative charge here, not three, one negative charge because it has combined the glycerol molecule. So it is a polar molecule because it is negatively charged, and it attracts water molecules which are also polar molecules, right? So it is a hydrophilic head or water-loving part of the phospholipid molecule. And then the fatty acids form the tails, which are hydrophobic tails, water-fearing or water-repelling parts. So the fatty acids are not charged at all, they are neutral, they are non-polar, they don't have uneven distribution of charges, so they are generally neutral, and they are called the hydrophobic tails. Right? So they do not attract water, they repel water instead. Now, for those who want to understand more clearly, you can look at this advanced information here, which will help you to understand how the structure of the phospholipid molecule is built up. Okay, so here we have the phosphate group in the hydrophilic head, PO4, negative here. And this phosphate group is attached to R. R here stands for the group that contains carbon and hydrogen. Okay. Then here below is the glycerol molecule that's attached to the phosphate group on top here and to the fatty acids below. Okay. So we can see the hydroxyl group has reacted with the carboxyl group here and then form the bond here. Alright? So that's the glycerol, part of the glycerol molecule, right? And then below we have the fatty acid, which form the tails, okay, the two tails. So one of the fatty acids is a saturated fatty acids without double bonds in its structure, whereas the other fatty acid is an unsaturated fatty acid and it has a double bond here, so it forms a band. All right, the tail forms a band that curves outwards like that. Okay, so this part is the hydrophilic head, and the one below are the hydrophobic tails, the two hydrophobic tails. You do not need to memorize the molecular structure here, all right? But this will help you to understand the structure of the phospholipid molecule better. The second type of lipid are the waxes. Okay, so wax, the structure of wax. It is formed when one molecule of alcohol combines with one molecule of fatty acid. Characteristics of wax. It is waterproof. That means it does not allow water to pass through it. Function. The wax forms the waxy cuticle on leaf surfaces in plants to reduce water loss from leaves by transpiration. Okay, so on a hot day, the water from the cells below, from the epidermal cells and the palisade cells, may tend to evaporate through the epidermis and the waxy cuticle. But the waxy cuticle will prevent water from evaporating through it, okay, from coming out through it, okay, because it is waterproof. So it will reduce water loss from leaves by transpiration or evaporation of water. Secondly, in animals, like in humans, sebum is produced, which is an oily substance that will coat the skin, and it contains wax. So sebum helps to coat and protect the skin from dehydration and also from bacterial infections. It is produced by the sebaceous glands or oil glands in the skin. So this diagram shows the molecular structure of wax. We actually don't have to memorize the formula, right? The chemical formula, but it's just to help you understand the structure of wax and how wax is formed. So wax consists of one fatty acid molecule that has combined with one alcohol molecule. Okay, so here is the fatty acid. It has the carboxyl group, which is COOH, right? And this is the alcohol. So alcohol molecule has a OH group here, hydroxyl group. So when the two molecules combine, one molecule of water is eliminated, and the fatty acid combines with the alcohol to form the wax. So this is the fatty acid component, and this is the alcohol component, okay, joined by this oxygen atom here. Let's go on to steroids. So steroids are lipids that do not contain any fatty acids at all, right? Now examples are cholesterol and some types of hormones, not all types, okay? Because some hormones are actually made from proteins, and other hormones may be made from steroids or other organic substance. Now, cholesterol is the most common steroid and is also produced in the human body by the liver. Functions is very important because cholesterol is needed as a precursor, that means as the main substance to build the hormones, such as testosterone and estrogen. That means before testosterone and estrogen can be formed, you must have the cholesterol molecule. And then from there, it will be able to form the testosterone and estrogen molecules. Notice that estrogen, we put an O here, textbook. For this chapter, the textbook misses out the O, just ES, okay? But actually it should be OES if we use the British spelling, okay? So in another part of the textbook, on the topic of hormones, you'll find that they add the O here at the beginning, estrogen, what estrogen. So another function of cholesterol is that it's a component of the plasma membrane, as we will find out later, and it helps to strengthen the plasma membrane and also make it more flexible. Okay. Now, lastly, cholesterol is used to synthesize bile salts, which are used to emulsify fats during digestion. So the bile salts will break up the lipids of fats into tiny droplets for faster digestion by the enzymes like lipase. Okay, so cholesterol is used to synthesize bile salts in the liver for fat digestion. Now, hormones. So we are going to discuss 3 hormones here. These hormones are made of steroids. They are steroids actually. Testosterone is the male sex hormone produced by the testes and is used to stimulate growth of muscles. That's one of its functions. Estrogen is the female sex hormone that's used to stimulate the repair of the lining of the uterus. All right, the repair of the wall of the uterus for to prepare for pregnancy. And then progesterone is another female sex hormone which stimulates the development of the lining of the uterus during the menstrual cycle, which we will discuss later on in another topic. So all these hormones are made of steroids, or they are steroids themselves, okay, apart from cholesterol. Secondly, let's discuss the importance of lipids in cells, and our learning standard is we must be able to justify the importance of functions of lipids in cells and in multicellular organisms. So, firstly, fats act as an energy reserve for humans and animals. So, fats act as an energy reserve or energy store for humans and animals. Now, in case a person doesn't eat enough carbohydrates and doesn't have enough glucose for oxidation to produce energy, all right, then fats will be the next source of energy. All right, so fats can be oxidized too to produce energy for activities, cellular activities and other activities of the organisms. Secondly, fats act as a liner to cushion and protect internal organs. Here we see the heart. It is actually covered by a layer of fat. Now, these yellow parts here are actually the fats, and the fats will surround the heart, okay, and if a person suffers from physical shock or a fall, then the fats will be able to cushion the heart and other internal organs to prevent injury. Thirdly, fats form a layer of adipose tissue under the skin, as shown here, the yellow part here, to act as a heat insulator for animals, meaning that it can prevent heat from escaping from the bodies of animals and humans, so it reduces heat loss from the body. So the thicker the layer of adipose tissue or fat, then the more effective is the heat insulator in the body to prevent heat loss from the body, especially in cold weather. Fourthly, wax is an important component in waxy cuticles that cover the epidermis of leaves, and it helps to reduce water loss from leaves by transpiration. So the waxy cuticle is the uppermost layer on the leaf, and it's a shiny layer. All right, that's why leaves look very shiny in the sunlight. So this waxy cuticle contains wax, which is waterproof. So it can help prevent water loss from leaves through evaporation, okay, from the cells through the epidermal layer, and then the water vapor will not be able to go past the waxy cuticle because the wax is waterproof. So this will reduce water loss from leaves by transpiration or by evaporation of water. The next function is number 5. Wax is an important component of sebum, and sebum is an oily substance secreted by the skin that contains wax. So this sebum will coat and protect the skin from dehydration and bacterial infections. As seen in the picture here, this is the structure of the skin, and here we have the gland just beside each hair. There's a gland called the sebaceous gland or oil gland in the body. So these sebaceous glands secrete the sebum, which is the oily substance, that can help to coat the skin and protect the skin from dehydration. Alright, so the sebum contains wax in it also. So plants produce the wax, and the wax is found in waxy cuticle. For humans also, wax is produced, and it is an important component of sebum. Okay, so for someone who always washes his or her hands, the sebum layer will be washed off, and then the skin will look very dry and dehydrated, like lacking water, right? So sebum is important to help protect our skin from dehydration, and it contains wax. Number 6, glycolipids. So glycolipids are another type of lipids, right? And glycolipids are actually lipids that contain a carbohydrate chain attached to the hydrophilic head, the red part here. So they are found in the plasma membrane, all right, inside the phospholipid bilayer, but the carbohydrate chain here is exposed, all right. So what is the function of glycolipid? So glycolipids ensure the stability of the plasma membrane that surrounds the cell by forming hydrogen bonds with the water in the environment. And secondly, they help in cell identification process. Now, what is cell identification process? So the glycolipids have the carbohydrate chains that act as markers to help the cells identify each other, so that the cells know which cells belong to the body, so that the white blood cells will recognize their own cells from the body and they will not attack them. But the white blood cells will be able to identify cells, foreign cells that are not from the body without those markers or glycolipids on top. Then they will be able to attack the bacterial cells and so forth. So glycolipids help in cell identification process so that cells can identify each other. Alright, cells of the same body can identify each other. Okay, so here you see the carbohydrate chain. This is a cross-section of the plasma membrane, and the lipid head is here, the hydrophilic head. So this whole thing is the glycolipid molecule. Next, cholesterol. So cholesterol is also found embedded in the phospholipid bilayer. Okay, so what's the function of cholesterol? As shown here, this is cholesterol, right, which is found inside the phospholipid bilayer. It makes the phospholipid bilayer stronger and more flexible. All right. And secondly, cholesterol is used in the synthesis of the steroid hormones such as testosterone, estrogen, and progesterone, which help to regulate body functions, especially related to reproduction. All right. So there are two types of hormones, or a few types of hormones. One type is are the protein hormones, hormones made from protein. Another type are the steroid hormones, okay, such as testosterone, estrogen, and progesterone. Lastly, the lipids in our diet that we eat help to dissolve fat-soluble vitamins A, D, E, K, and help in their absorption in the small intestine. So if you are taking any foods containing vitamins A, D, E, K, remember to include some lipids in the diet too, in the meal, so that these vitamins can dissolve in the lipids and then they can be well absorbed into the small intestine. Okay? For example, if you're eating carrots that contain vitamin A, add some lipids like olive oil into the salad that contains the carrots, right? So this will help the vitamin A from the carrots to be well absorbed, efficiently absorbed into the small intestine. Next, we're going to discuss a food test to test for the presence of lipids. This is the third learning standard. So the food test for lipids is important to remember, and it may even be asked in the paper 3 practical exam where you'll be given a food sample to test, right? So it's important for us to know the procedure and know how to test for the presence of lipids, fats, or oils in a given food sample. Right, so this test is called the emulsion test, okay, for presence of lipids, fats, or oils, or the ethanol emulsion test. Let's look at the procedure. Firstly, add 2 ml of ethanol to 1 ml of the liquid food sample in the test tube. So if it's a liquid, you just get 1 ml of the liquid food sample and put it in the test tube, then add 2 ml of ethanol. If it is a solid food sample, then you have to crush the food sample using a mortar and pestle first of all, and then get 1 or 2 spatulas of this crushed food sample and put it into the test tube first. Okay, then add the 2 ml of ethanol. Next, shake the test tube for 2 minutes to ensure that the ethanol dissolves any lipids in the food sample. So ethanol, which is an alcohol, can dissolve the lipids or oils or fats. Okay, but water cannot dissolve this any lipids or oils or fats, but ethanol can. So after you shake the test tube, next you add 2 ml of water, the same volume of water, okay, as the volume of ethanol used, right? Same volume. So add 2 ml of water to the test tube and then shake again. Okay. Now, at the end of this, wait a while and see if a milky white emulsion is produced. So you may see a layer like milk at the upper part of the test tube. So this is called the milky white emulsion. If there's a milky white emulsion, it proves that lipids are present in the food sample. But if the mixture is clear, that means no lipids are present. Now, what is this milky white emulsion actually? So if lipids are present, the lipids will dissolve in ethanol first of all, and they will form tiny droplets when the mixture of ethanol and lipids are poured into are mixed with the water. Okay, or when water is added to the ethanol and lipid mixture. Okay, so what happens is that the lipids will form very tiny droplets, and they will be suspended inside the water alcohol mixture here. Okay, dispersed and suspended like a suspension. So this is called the milky white emulsion. Okay, to prove that lipids are present in the food sample. And you can also watch a video on the emulsion test for lipids. Okay, just click this website here or address here, right? And watch the steps taken to test for the presence of lipids or fats using the emulsion test. Right, that's all for this lesson. Please do share, like, and subscribe for more videos that are beneficial to you. So, thanks for viewing and do take care. Goodbye for now.